Okay, for part 2 here, we're going to talk about sea slugs. How exciting will that be for you, huh? So with sea slugs, you need to turn to your book, page 517 and 518. Sea slugs technically are given the term, are called aplesia. And actually, if you turn to page 517 at the very top, it says the guy who's doing research on this, and the guy we'll be talking about is Eric Kandel. He's actually won Nobel Prizes for his work with sea slugs. And it'll be the same idea we saw in the previous presentation. We're interested in what's going on with learning and memory things here per se, and trying to understand the neurobiological roots for what's going on here. So a little basics as far as sea slugs and how they actually work. In the first situation, if you turn actually to page 518, if we actually take in our simple procedure here, there's a tail. A sea slug has a tail, and a sea slug has a gill. And if you take actually, if you're a researcher and you deliver a sharp electrical stimulation to the tail, what normally happens when you do an electrical shock to the tail is you'll see the gill withdrawal. So if you shock the tail, the gill will withdraw because it's kind of a defensive mechanism in terms of the organism. So that's the simple basic procedure. You shock the tail and you see the gill withdraw. Now, the second part of this is also very interesting. Normally, if you actually go to an area called the siphon or the mantle shelf, I believe it is, but we'll actually call it the mantle shelf. If you actually go in there and normally just do a light stimulus to the mantle shelf, usually you don't see any sort of gill withdrawal response. Usually you just see basically nothing. It's like you're touching the mantle shelf and the gill doesn't withdraw. But what's really interesting in our procedure is what actually happens when you do the following. If you actually take the tail and deliver a sharp electrical shock to the tail, and right around the same time, if you pair that up with touching just the mantle shelf, maybe even touch the mantle shelf right before you do a shock to the tail, what you actually find is, of course, you'll see a big gill withdrawal reflex. So actually, let's set this up again. Let's say you take the mantle shelf in phase 2 here, and in phase 2 you just touch the mantle shelf first, and then you do the sharp tail electrical shock. You'll see the gill withdraw. But what's really interesting is kind of the last phase of the experiment. Once you do the pairing here between just touching the mantle shelf and the sharp electrical shock, in phase 3, if you go back and you just touch the mantle shelf in phase 3, all of a sudden the organism, the aplesia, will have a vigorous gill withdrawal response. So the idea is, in phase 1, where you just touch the mantle shelf, you didn't see any gill withdrawal. In phase 2, when you touch the mantle shelf first and then you shock the tail, you see a strong gill withdrawal mechanism. But then in phase 3, what seems to be new here is when you just touch the mantle shelf in phase 3, the organism has a strong gill withdrawal reflex. So this is many times called by aplesia, or by Kandel I should say, this is called sensitization. Something became sensitized, whereas now you're responding to something simple, just touching the mantle shelf, with a strong gill withdrawal mechanism. So that is the kind of classical conditioning thing happening again with sea slugs here. It's called sensitization, and you can read more about it in 517 and 518. The question for this study was very similar to the previous one that we looked at. Can we actually find out what's going on neurobiologically where before you would just touch the mantle shelf and nothing would seem to occur for the gill withdrawal, and now when you touch the mantle shelf after the conditioning, all of a sudden you get a strong gill withdrawal mechanism. The picture on 518, figure 1412, explains it, and we're not going to go into a ton of detail here, but if you look at that particular picture, you can see that if you actually deliver the electrical shock, the idea is there's a pathway for that electrical shock, and notice there's another pathway for the weak touch to the mantle shelf. What seems to occur is if you follow those pathways, that now when you touch the mantle shelf, if you follow the pathway there, you can see there's a neuron that has a synapse to the motor neuron from the mantle shelf. It seems there's an interneuron that actually has a synapse with the mantle shelf presynaptic neuron. So there's actually a connection with an interneuron. So it seems that electrically stimulating that strong pathway from the tail actually facilitates a neuron to have a synapse right on that mantle shelf synapse, so that now the mantle shelf synapse becomes very, very strong because of the connection with the electric shock tail synapse. So the idea is the electrical shock stimulus pathway neuroanatomically has a synapse with the presynaptic area of the mantle shelf sensory neuron. So the idea is there does seem to be a connection here. The idea is there's kind of overlapping neurons, and the direct pathway, the tail shock pathway, actually causes the other pathway to become strengthened because of the overlap here, and thus now touching the mantle shelf leads to a vigorous response because of the tail shock pathway. I know I haven't done a brilliant job with explaining this to you. If you want to read a little bit more, obviously, in your textbook to kind of summarize that, but that's the idea. Once again, much like the first presentation, we see something here called sensitization, which is an emotional response. Is there a neurobiological correlate even in these simple sea slugs? And the answer appears to be yes. So thank you very much for your attention, and that'll do presentation number 2.